This is DNA Replication, a semi-conservative story of love by Michael Wong. Scene 1. Nucleotides are made of nitrogenous bases, deoxyribosyl sugar, and phosphate group. There are base pairs called adenine, thymine, guanine, and cytosine. Between these, there is a bond called the hydrogen bond. It is quite weak. Complementary pairs form between these base pairs, and such that guanine matches with cytosine and adenine matches with thymine, so GCAT. Phosphate sugar backbone, it basically just means that it is composed of the phosphate group and the deoxyribosyl sugar from the nucleotides. The backbone connects together through a phosphodiester bond. Double helix is just the structure of the DNA, and it's just two strands of DNA that kind of intertwines together. Next, scene 2. The helicase begins by in the replication fork. Well, at the place where the replication fork is at. It is an enzyme that separates the two DNA strands by first unwinding the double strands and then breaking up the hydrogen bond. Scene 3. The RNA primase is an enzyme that synthesizes a short RNA primer. The RNA primer then attracts the DNA polymerase III and then initiates the process of replication. Scene 4. DNA polymerase III starts off the replication process as it attaches to the RNA primer. It attaches to the 3' end and moves towards the replication fork in the leading strand. In the lagging strand, however, it moves away from the replication strand. Scene 5. DNA ligase is basically just the glue that is used to put together the Okazaki fragments during replication on the lagging strand. DNA polymerase I, it just removes the primers and replaces them with DNA and DNTPs. The leading strand is just a strand in a double strand that goes from the 5' end to the 3' end. And on this strand, the polymerase goes towards the replication fork. The lagging strand, on the other hand, is a strand opposite of the leading strand. It moves from it goes from a 3' end to the 5' end, and the polymerase goes away from the replication fork. Okazaki fragments are the small little strands which are made on the lagging strand because the polymerase goes in the opposite direction from the replication fork, so it is a discontinuous kind of motion. Semi-conservative replication, it really just means that one strand is kept while the new one is being made, and then after it is created, they are paired together. Yes, thank you.